There are more chickens on Earth right now than there are humans. Roughly 25 billion of them, spread across every inhabited continent, in coops behind suburban houses, in massive industrial barns, in backyard flocks in villages that have kept chickens for 1,000 years. They come in hundreds of breeds. The towering Brahma with feathered legs like a costume. The tiny Silkie with fur instead of feathers. The Leghorn that lays an egg almost every single day. The Cochin, the Orpington, the Rhode Island Red. And every single one of them, every chicken alive on Earth today, traces back to a wild bird species that most people have never seen and most people have never heard of. The question of where chickens came from sounds like it should be simple. Chickens look like small ground-dwelling birds. Pick any pheasant family species and the resemblance seems obvious. But when researchers actually ran the genetic comparison, sequencing the genomes of domestic chickens from farms across the world and testing them against every candidate wild bird species, the answer took decades to settle, and it was more complicated than anyone expected. The pheasant family, which is where the chicken belongs, contains dozens of species spread across Asia, from the Himalayan foothills down through the jungles of Southeast Asia. Peacocks are pheasants. Partridges are pheasants. So are the ring-necked pheasants that show up in American hunting preserves. Any one of them could plausibly be mistaken for an early ancestor of the barnyard chicken. Researchers tested domestic chicken DNA against this full range, and most of the family was eliminated almost immediately. The genetic distance was too large. The physical similarities that made these birds look related to chickens turned out to be a case of shared ancient ancestry, going back millions of years, not a recent domestication event. The comparison narrowed quickly to a smaller genus called Gallus, the junglefowl. There are 4 living species in this genus: the gray junglefowl of southern India, the Ceylon junglefowl, found only on the island of Sri Lanka, the green junglefowl, isolated on the island of Java in Indonesia, and the red junglefowl, which ranges across a huge stretch of South and Southeast Asia from northeastern India through Myanmar, Thailand, Vietnam, and southern China. All 4 species look strikingly similar to a domestic rooster. All 4 have that same silhouette: the comb, the wattles, the arched tail feathers. If you saw one in the wild without context, you would likely assume you were looking at an escaped chicken. The Ceylon junglefowl was eliminated first. It is confined to Sri Lanka and shows a genetic signature distinct enough from domestic chickens worldwide that it could not be the primary ancestor, though its geographic isolation made it an interesting outlier in the pheasant family generally. The green junglefowl of Java followed. It is the most visually distinct of the 4, with a different call and different plumage patterns, and the genetic distance from domestic chickens was significant enough to rule it out as a founding population, even though localized interbreeding with domestic birds has been documented on Java in more recent centuries. That left 2 candidates, and this is where the story got interesting. The gray junglefowl of southern India was a strong contender. Genetically, it sits close to domestic chickens, closer than either the Ceylon or green junglefowl. But the primary genetic signature running through domestic chicken populations worldwide, the backbone of the modern chicken genome, matched the red junglefowl most closely. Specifically, a population of red junglefowl from the region spanning what is now northern Thailand, Myanmar, and southern China. That did not fully close the case, though, because of one detail that puzzled researchers for years. Domestic chickens worldwide carry a gene responsible for yellow skin, the trait that gives supermarket chicken its pale yellow color rather than the white or gray skin found in most wild pheasants. Red junglefowl do not have this gene. Gray junglefowl do. When researchers traced the yellow skin gene specifically, it pointed straight back to the gray junglefowl of India, meaning that at some point after the initial domestication, domestic chickens and wild gray junglefowl interbred, and that hybridization left a permanent, traceable mark on every yellow-skinned chicken eaten on Earth today. The modern chicken is not the descendant of one wild species. It is the descendant of one primary wild ancestor, the red junglefowl, carrying at least one significant genetic contribution from a second species picked up somewhere along the way. The timing of the original domestication event has itself been a moving target. For decades, the Standard account placed chicken domestication in the Indus Valley roughly 4,000 years ago, spreading from South Asia outward. But as ancient DNA techniques improved and more archaeological sites were reanalyzed, that story fell apart. Bones that had been identified as early domestic chickens in China and elsewhere turned out, on closer inspection, to belong to other bird species entirely: pheasants, quail, or birds too poorly preserved to identify with confidence. When researchers stripped out the misidentified remains and focused only on bones that could be confirmed through direct dating and genetic analysis, the earliest solid evidence of domestic chickens shifted dramatically, both in time and location, to Southeast Asia, in the region of what is now Thailand, roughly 3,500 years ago, thousands of years later than earlier estimates had claimed, and in a different place entirely. What actually drove the domestication is its own story, and it complicates the tidy image of early farmers simply taming a convenient bird for meat and eggs. Red junglefowl in the wild are cautious, alert animals, difficult to approach and quick to flee. The current best explanation for how they ended up living alongside humans involves agriculture, but not in the way you might expect. As rice farming spread through Southeast Asia, it created something the surrounding forest did not have: large, concentrated, reliable stores of grain. Red junglefowl, like many ground-feeding birds, are drawn to grain. The birds that were bold enough to feed near these new rice fields and human settlements gained access to a food source unlike anything available in the surrounding jungle. Over generations, the population living close to farming communities became incrementally more tolerant of humans, while the population deeper in the forest remained wild and wary. This mirrors, almost exactly, the pattern discovered in the domestication of the cat, a wild animal drawn in by a byproduct of early farming, gradually settling into human company not because it was captured and bred, but because it chose an environment that rewarded staying close. From that founding population in Southeast Asia, the domestic chicken spread with extraordinary speed, considering how limited early trade and travel networks were. Genetic and archaeological evidence shows chickens reaching the Indian subcontinent, the Middle East, and eventually the Mediterranean world within a few centuries, likely moving along the same overland and maritime trade routes that carried spices and textiles. By the time of ancient Egypt, chickens were present, but still relatively rare and expensive, more a novelty animal than a staple food source. It would take the Roman Empire, with its extensive road networks and appetite for standardized agriculture, to turn the chicken into the mass food animal it is today. Roman writers documented specific breeding practices for egg production and meat, some of the earliest evidence of deliberate selective breeding applied to poultry. The genome itself tells a story of extremely rapid, extremely intense selection, more intense in some ways than almost any other domesticated animal. Unlike the house cat, which barely changed from its wild ancestor because no one was selecting it, the chicken has been reshaped dramatically, and increasingly so in just the last century. Modern commercial broiler chickens, the breed raised specifically for meat, reach market weight in under 7 weeks, a fraction of the time it would have taken a chicken even 50 years ago. That speed is not incidental. It is the result of one of the most aggressive selective breeding programs ever applied to a domestic animal, concentrated overwhelmingly in the decades since the 1950s. Genes controlling growth rate, muscle mass, and feed efficiency have been pushed to their biological limits, and researchers studying the modern broiler genome have found signatures of selection so strong and so recent that they are almost unmatched anywhere else in domesticated animal genetics. And yet, underneath all of that engineered growth and all of those specialized breeds, the genetic backbone remains the red junglefowl of the forests along the Thailand-Myanmar-China border, occasionally streaked with the yellow skin legacy of a gray junglefowl from India that, at some point thousands of years ago, crossed paths with a domestic flock and left its mark permanently on the species. The red junglefowl itself still exists in the wild today, growing rarer as habitat disappears and as wild populations increasingly interbreed with free-ranging domestic and feral chickens, a process that is slowly erasing the genetic purity of the very ancestor that gave rise to 25 billion of its descendants. The comparison of every wild bird species on Earth to the domestic chicken produced an answer that once found seems almost too ordinary. Not a mythical bird, not a species long extinct, but a shy, ground-dwelling forest bird from Southeast Asia that a few bold individuals, drawn by spilled grain near a rice paddy roughly 3,500 years ago, decided was worth the risk of coming closer. Every egg cracked into a pan this morning, every rooster crowing at dawn anywhere on Earth, carries forward a decision made by a handful of wild birds in a Southeast Asian forest. Long before anyone thought to write any of it down,